Hello, my name is Anna and I'd like to show you today how to separate Legionella cells from a water sample with the rqmicro CellStream instrument and the rqmicro Legionella pneumophila serogroup 1 kit. In a first step, I'm filtering the water sample with a standard filtration unit. I am then transferring the filter that rqmicro provides with the kit into a Falcon tube that contains 3 ml of buffer. By vortexing, I resuspend the cells in buffer. Now I will add 2 types of antibodies produced by rqmicro to my sample. This mix contains antibodies bound to magnetic particles. They will bind to the target cells and thereby enable the immunomagnetic separation. I'm also labeling the Legionella with antibodies bound to a fluorescent dye. Thereby I can quantify them with the flow cytometer after the immunomagnetic separation. Finally, I incubate the samples for 30 minutes at room temperature, so the antibodies can bind to the target cells. I then load my sample or samples into the disposable cartridge. Then I put the cartridge into the cartridge chamber of the CellStream and start the separation process. The sample passes through a narrow channel in the cartridge. During this process, a magnet is lowered down and thereby diverts Legionella cells which are bound to magnetic particles. At the same time, more than 95% of the competing flora originally present in the sample is directed to the waste fraction. Due to a washing step, the sample purity reaches up to 99%. At the end of the process, the magnet is raised and the Legionella cells are collected in clean buffer in a final volume of 1 ml. In this last step, I'm quantifying the target cells with a flow cytometer. If you do not work with flow cytometry, you can also analyze your sample by PCR, fluorescence microscopy, or simply by adding a specified volume of the sample onto a Petri dish. And that's it. Thanks very much for watching, and if you have any questions about the procedure, please don't hesitate to ask me or my colleagues from the rqmicro team. Further information you can also find on our website rqmicro.com.